everyone. Welcome to the Invasive Mussel Collaborative webinar, Case Studies in Manual Removal of Invasive Mussels. My name is Erica Jensen. I'm the Aquatic Invasive Species Program Manager at the Great Lakes Commission. And on behalf of the Invasive Mussel Collaborative, thank you for joining us today for our webinar. For those of you not familiar with the collaborative, the Invasive Mussel Collaborative was convened in 2015 to identify opportunities to advance scientifically sound technology for management and control of invasive drycenid mussels to produce measurable ecological and economic benefits. If you'd like to know more about the collaborative, we encourage you to visit our website at www.invasivemusselcollaborative.net. Today's webinar is part of an ongoing series hosted by the Mussel Collaborative. The purpose of these webinars is to facilitate learning and information sharing on topics of common interest within the invasive mussel community. It will be recorded and archived along with past and future webinars on the Mussel Collaborative website. On today's webinar, we will explore the decision-making processes leading to manual removal of invasive mussels, operational and logistical considerations and constraints, and lessons learned from past removal efforts and an ongoing removal effort. We're pleased to have speakers from the Rensselaer Polytechnic Institute, the University of Georgia, the National Park Service, and the University of Wisconsin-Milwaukee joining us to share their experiences today. While we always strive to minimize technological issues during these webinars, we realize that some may arise and we apologize in advance. Toward this end, we'd like to cover a few logistical items that may help before we get to the presentations. The audio for this webinar is automatically streamed through your computer speakers once you join the webinar, but you also have the option to dial into the webinar using a telephone. If you'd like to change your audio to a telephone line, you can select Switch Audio to My Phone from the banner at the bottom of the screen and click Connect from the pop-up screen. A second pop-up screen will list the number to call into as well as a conference ID number that you can use to connect to the webinar. If you are experiencing technical difficulties at any time, you can send a message through the webinar chat box located in the lower left-hand corner of the webinar window. To view the presentations in a full screen format, you can change your screen layout to Content View. To change the layout, simply select the layout icon in the upper right corner of your screen indicated by the red circle on the slide and choose Presentation View or Content View. In order to minimize the potential for interruptions and background noise, we have automatically muted all attendees' audio lines. Attendees will remain muted throughout the webinar. That being said, we do encourage and will accept written questions from attendees at any time during the webinar. If you have a question, you may type it into the chat box signified by the chat bubble icon on your webinar window. Questions may be submitted at any time, and depending on timing and how on schedule we are, we may answer one or two questions following each presentation, or we may hold the majority of questions till the end after all the presentations have been given. We always do our best to take all questions or comments during the webinar, but if you need to leave the webinar before we've gotten to your question, we encourage you to review the webinar recording that will be posted on our website to hear the question and answer. So those are all of our housekeeping items for today. So right now I'd like to move to introduce our first presenters. They are Dr. Marc Frischer and Dr. Sandra Nierzwicki-Bauer. Dr. Marc Frischer is a professor of marine sciences at the University of Georgia's Skidaway Institute of Oceanography. He received his PhD from the University of South Florida in 1994 in marine sciences, specializing in marine microbiology, and completed a postdoctoral fellowship at the Rensselaer Polytechnic Institute, where his work with zebra mussels began. Professor Frischer's research centers on the development and application of molecular biology tools in marine and aquatic ecology, including molecular approaches for tracking, detecting, and quantifying invertebrate larvae such as zebra and quagga mussel veligers. Dr. Sandra Nierzwicki-Bauer is a professor of biology and the current associate director of the Darren Freshwater Institute Field Station on Lake George at Rensselaer Polytechnic Institute, after serving as the director for over 20 years. He's received a PhD in microbiology from the University of New Hampshire and was a postdoctoral fellow at the University of Chicago. A former department chair of biology and interim dean of the RPI School of Science, Dr. Nierzwicki-Bauer's work focuses on molecular studies of microbial and eukaryote communities in lakes and rivers, invasive species such as zebra mussels and Asian clams, and water resource management and environmental remote sensing. So thank you and welcome to both Dr. Frischer and Dr. Nierzwicki-Bauer. At this time, we will hand over presentation control to both of you.
Very good. All right, I think I'm in charge.
You are.
Okay. And you guys see the first title slide?
Yes.
All right. Well, thank you very much. This is Marc Frischer for the introduction and for the invitation to be here. Both me and Sandra are very excited and pleased to be able to share with you what is now a few years ago our experience sort of with the invasion of Lake George by zebra mussels and our continued efforts to keep them out. So I will move on. All right, so Lake George, for those of you who may not be familiar with Lake George, is in the Adirondack Park in upstate New York. It's a 6 million acre state park, a wilderness area, but there are many areas in it that have been populated since colonial times, and so there is still some human presence there. And Lake George has been a long-time destination for a lot of people for recreation and for all sorts of things. So there are considerable human occupation despite its being in this wilderness area. We know it's a large, relatively deep meso-oligotrophic lake, so fairly low nutrient. We do have a number of invasive species that are in there or keep trying to get in. These are the ones that we know about, at least, in no particular order of when they came. But they're in different stages of the invasion curve. So the macrophytes, the Eurasian water milfoil and the pondweed, those are fairly well established at this point, and we're in control mode, as is Asian clams, and we're still sort of trying to fight off zebra mussels and the spiny water flea. There's a lot of access and a lot of sources of potential introduction of invasive species to Lake George, and so we have a fairly extensive and active aquatic invasive species prevention program that includes boat inspections. And so this is just a Google map of locations where boats have entered and had some sort of invasive species on it. You can see that there's quite a lot of it. We're attracting a lot of potential invasives this way. And we'll get back to talking about that program as we go through this talk. So in terms of zebra mussels, we first detected them, and not as zebra mussels, but as larvae in 1995. And based on where we detected them and when we detected them, we conclude that they probably are being transported by boats or maybe construction or dredging equipment that's being transported and brought into the lake from somewhere else. And we also know a little bit about the water, and we know actually quite a lot about the lake itself, and it's based on its water chemistry, particularly its low calcium content, is not the most hospitable place for zebra mussels. So this is just some of the risk assessment based primarily on water chemistry and calcium and pH. And you can see in the, I don't know if I have a pointer. Oh, I do have a pointer. Talked about this. Which was this one? Hopefully you can see my pointer now. This is the model that we were using at the time when we were making these detections. Lake George is right on the borderline of being a place that zebra mussels could be, and that's sort of confirmed by additional data being added to these models in this EPA model. So it's a low-risk place, but I won't say that it's without risk. And we were quite vigilant. And it sort of paid off. Although we had been detecting larvae veligers, zebra mussel veligers, in the lake since 1995, we had not seen, despite our surveys, we had not seen any adult zebra mussels there until 1999, and not by us, but by volunteer divers who were doing a lake trash cleanup in some of the more populated areas. And they found, while they were cleaning up trash, they said, this doesn't look like it should be here. And they, because we had been talking about this in the community, recognized those as zebra mussels and immediately told us about it. And so we went out and started to investigate. Okay. So this is a nicer time of year when this picture was taken, but this is the area where we found those zebra mussels. You can see it's fairly crowded, populated place. This is in the village of Lake George. It's at the southern end of the lake and is the most populated place. It's a big sort of tourist destination only in the summer. And we found them in this area that's delineated by this yellow line that I think you can see there. There's a boat launch right over here. So in our initial surveys, the first thing that we did was try to see to what extent the infestation invasion might have been, and so we sent some divers in there to survey and look around, and we found that it seemed to be a fairly limited area with a clear concentration in one actually really relatively small area that we have labeled here as called hotspot. And so after our very initial surveys, we delineated this area and we set up quadrants and to do a systematic evaluation of what we had there. And these are our results from that initial survey. That we see that most of the animals that we were pulling out came from these middle sections where we have the hot spot, this section 4 and 5, and it dwindled off as you went in both directions. Okay. And so although we did find a few out here, they were very far and few between, and we felt fairly confident that we could say that the infestation was restricted to this area. I should also say that, like most of Lake George, this area, the bottom, the benthos, is sand and mud and silt with some rocks and some hard structure in there. And so it wasn't always that easy to see these animals. And so it's quite a laborious effort to go in there and make sure you're really getting rid of them. Okay. And this is a size histogram frequency look at what we found from that first survey, and it suggested, you see, it looks like we had one cohort, not multiple ones, which was good because we think that this is a single population. And based on the size and the estimated growth rates of dracenids, although we didn't have growth rates in Lake George, we estimated that this invasion was a result of a introduction in 1997 or 98 that was followed by one successful recruitment. So there's only one real cohort here. And that was, it wasn't good news that we saw them there, but it was good news with respect to where we thought we were in terms of the invasive invasion cycle. We think that this is one of these times that we were lucky enough to really catch them at the earliest of their introductions. We had been sort of watching and paying attention, and this site actually was on our radar because it's a fairly active place where boats are put in and pulled out. And so we're pretty sure that this might have been the first introduction into Lake George. And so that gave us a lot of options, and even considering the one of eradicating. And these are the options that we considered looking into. We could do nothing. We knew that the water chemistry was not very conducive to these animals and that they might not reproduce or reproduce very well, and that maybe the problem would just go away. That was certainly part of our discussion. We thought about putting in, isolating the area with cofferdams, and ultimately decided that would cost too much and would be a lot of permitting. Remember, this is a state park. It's quite highly regulated. We would have needed a lot of construction permits and that kind of thing. We considered some kind of chemical treatments, and remember, we're back in 1999, so we didn't have as many treatments available at that point. We probably wouldn't have even been able to get a permit because we just wouldn't have had enough data to demonstrate that wouldn't have other effects. We considered benthic mats, but again, the bottom is the topography of it. It's not just a flat meadow, and it might be difficult. We thought it would be fairly difficult to put them in place, and then especially when the docks went back in, because they take those docks, the boat ramps, in and out and on an annual basis, or some of them, it might be hard to keep them there. So even though we've been using and had been using benthic mats to help control the macrophyte populations, we didn't know if we could make that work for the zebra mussels. We also started getting a little crazy and thought about some kind of heat treatment, maybe taking a steam gun and down there and spraying the affected areas. Again, it was fairly contained, and we thought we might be able to do that, but nobody had ever really tried that in any kind of real-world situation, so we didn't know if it would work. And then when we really got down to thinking about, well, what would that involve, we started to get worried about diver safety and could we operate this stuff down there. I mean, we have a scientific diving program, but this definitely would have fallen into a sort of more professional diving program, and we were concerned about that. And that left us with the idea of hand harvesting them. We only needed a permit to work there, and we were ready to go with that. So that's how we ended up with this idea that we would try this hand harvesting, although we were certainly fully cognizant that it would be difficult to get every one of them this way. So our course of action at the time was to assess the problem and determine the population size and area of infestation. I've already talked about that. We started a program to systematically hand harvest them, to eradicate, with the goal of eradicating them. And then through the whole process, we've been trying to figure out why that happened and to determine really some more science about what happened, where did they come from, are there measures that we could put into place that would help prevent further invasions. So this is the site. We actively were eradicating for 8 years, from December when we first detected them through May through spring of 2007. Most of this work, we would visit the site twice in a year, in the fall after the tourist season was over and there weren't so much traffic there, and then in the spring before that started again. So we would visit the site those two periods to do our hand harvesting. And this figure just shows the first couple of years of that. After about 2003, we were really collecting very, very few animals. But you can see in this curve and these histograms again that both we were reducing the size of the population, that they were growing. They were growing in Lake George water for sure. And then it looked like pretty much we probably still had just that single cohort through the process. So this was all good news and very encouraging that we were having the impact that we had hoped to. And this is a figure that shows how many mussels we remove with respect to how much dive effort we put into the process. Fairly clear exponential relationship, and we, with sort of diminishing returns as we continued. Most of the animals, as I said, came out in the first couple of years. Okay. And then we reached some sort of asymptote, at least statistically. So if we had had a good idea of how many animals it would have taken, for example, science question could be how many do you have to get rid of before it no longer becomes a viable population? That's a question we still don't have an answer to. Right? We also, by estimating the sizes of the animals on each successive collection, were able to estimate growth rates, and that gave us that's how we calculated when we think the initial invasion occurred. So they grew pretty well on Lake George, a little bit slower than in the Great Lakes. Okay. And we also were able to do economic analysis about what it cost. It took us over those periods about 860 hours of diving in the water, but that you can times 4 that to get to how much shore support that the divers needed to make that, have to figure that in. We also did a lot of water collection and data analyses associated with this, which is also more than about double the actual hours in the water. And spent some time reporting and other activities. We managed to run this program with one full-time scientist and a small amount for other professionals that added in over the course of the program, and a whole lot of volunteers. So it was really a community effort to get involved in this and to eradicate them from Lake George. Our funding came from various grants, and initially we got some money from New York Sea grant, which is why we were to develop new technologies to detect larvae as an early detection strategy, and then we've had continued funding from several of these conservation organizations, including the Helen V. Freilich Foundation, the Lake George Watershed Conference, and the Fund for Lake George. And I think with that, I will hand over to Sandra.
Muted. Sandra, you're unmuted.
Did we lose Sandra?
No, can you hear me now?
Yes.
Yeah, we can hear you.
Perfect. And is my first slide up?
Not yet. We'll need you to share your desktop with that blue button at the bottom of the screen.
Oh, thank you.
Loading now.
Great.
All right, so if you just minimize the Skype window. Perfect. We're good to go.
Okay, thank you very much. I'm going to follow up on where Marc left off, as well as talk about continuing studies that we're carrying out in Lake George, which includes additional scuba removal of other locations that zebra mussels have been found. But let me start with ending Marc's proportion with what the good news was, in that there was no evidence of further spawning or recruitment at the Lake George Village site he was showing you, where over 21,000 adult zebra mussels were removed. As we've been going back there, because we had gotten out the large percentage of zebra mussels, we've been finding no veligers. We found no recruits or juveniles, and no small adults. So with that information, we feel pretty comfortable that in that one localized area, we have been able to, via scuba diving, eradicate the zebra mussels. Now, that doesn't mean eradicate the zebra mussels from all of Lake George, but at least we were able to, in that location, eliminate the presence of zebra mussels. So what this means is, with that success, we wanted to be sure that we had continued vigilance and initiated surveys at other locations, in particular marinas. These would be, obviously, our high-risk sites based on most likely modes of introduction. And so even to date, we carry out yearly marina surveys. The map shows the locations where we carry out those surveys, the marinas that we're looking at. We do several transects along the docks and the seawalls and the boat launches. As Marc had mentioned, luckily in Lake George, calcium concentrations are kind of borderline for zebra mussel growth. And in areas where subsequently we have found zebra mussels, we're very interested in what the calcium concentrations there are, because we could have niches or pockets in particular locations with elevated calcium that promotes the colonization and further growth of zebra mussels in those areas. When we do these yearly surveys, we remove all of the zebra mussels that we find. We measure them, we preserve them. We're trying to look at what is occurring within populations at different locations. But I should say all of the populations are quite small. This slide shows a timeline from 1999 to 2015. And as its animation occurs for each year, the new white dot that appears is the first time a location was found to have zebra mussels. And so there's some really good news here. As you can see, the Lake George Village site is where we had that largest population that we removed by scuba. At all of these other locations, we have also proceeded with removal of zebra mussels by scuba. However, once we get to 2011, since then, we have not found any additional locations, new locations with zebra mussels. And you can see that in many of these, the populations that were found and the mussels that we've removed are really pretty low. Probably, for example, in Boone Bay, this one is dropped off of a boat because there really was no population there. But yet we go back to all of these sites every year and survey them and remove any adult mussels that are present. In addition to us doing scuba at locations that are most likely modes of introduction, we respond to citizen reports. And we don't get a lot of citizen reports, but maybe 2 or 3 times per summer, a concerned citizen will contact us and say, I think I found a zebra mussel. We encourage them to take a sample and give it to us, or to take a photograph. But when this occurs within a week, we carry out a survey at that location and then determine what the next steps are. In many instances, there's banded snails, banded snails that people confuse with zebra mussels. But in some instances, when there's been a real concern, we'll do a survey and remove any zebra mussels that are found. In addition to looking for the adult zebra mussels, as Marc had mentioned, in 1995, we started a villager monitoring program, which still is ongoing. Shown in the map, we have 12 different locations where we visit bi-weekly throughout the summer months. We collect 200 liters of water, concentrate that sample, and then examine them with cross-polarized microscopy. If any zebra mussel villagers are found, then we go back and do a scuba survey for those. In addition to that, we had been doing recruitment monitoring. We did this from '95 until 2013. In this program, we went to 4 locations, and we would visit them twice a year. We essentially set up, as shown in the figure, a spat trap which has stainless steel plates to provide a surface for zebra mussel villagers to settle on and grow. It's interesting that with this approach, we did detect the occurrence of zebra mussels, or the introduction of zebra mussels, at one of our northern locations. However, since that time, since 2013, we've discontinued the program, mostly because the cost didn't justify the return, and we felt as though from our marina surveys as well as our villager monitoring, we were getting the necessary information that we required. So, as I'm sure all of you know, prevention is the key. Education and outreach is a huge part of that. Raising public awareness and knowledge in having citizens call us. In addition to that, though, education and outreach in Lake George and many other places has been increasing by establishing boat wash stations. These are free boat wash stations, but in Lake George, at least, they are mandatory. Marc had shown you a slide that where boats coming into Lake George are coming from. This is a different rendition of that, which specifically hones in on where did boats come from before coming into Lake George, which in this boat inspection program actually carried zebra mussels. And so you can see in the map there's about 19 locations or other water bodies surrounding us, and as far north as above Quebec City here, where boats coming in would have brought new zebra mussels and introduced zebra mussels, but because of these inspections, they were stopped. So there's about 25,000 inspections that are carried out annually. This just shows a snapshot of those results. So in 2014, the boat wash stations became mandatory. And this shows the results from 2014 to 2017. And you can see that through these inspections and boat washes, that there has been a prevention of 10 to 30 potential new introductions of zebra mussels per year into Lake George. I want to circle back for a second in terms of the presentation or the portion that Marc gave and what happened at Neptune's. And this, I think, can happen in general in Lake George. But clearly, in the location where the first zebra mussels were found, there was some elevated calcium concentrations. This could have been possibly due to construction project going on, through drought that we had or other means. There's some type of human introductions of the zebra mussels. In the southern site, we believe that it may have been due to construction of a boardwalk there. And they did some dredging, and it could have come in on heavy equipment. But obviously, the more traditional or routine route of introduction would be boats, which resulted in zebra mussels in Lake George. Going back to the photograph that Mike had shown and our drawing of the site, we did have elevated calcium there. There was a culvert, I'm pointing to it now, and it's shown in red, that had over 24 milligrams per liter of calcium. compared to the average in Lake George, which is below 12, as well as other lower concentrations throughout. So it kind of, in this particular instance, was a particularly good environment for those zebra mussels to survive there. So it was really good that we got to them early on. I wanted to mention just a couple of additional activities that we've been doing or have done to help us better understand as well as monitor and make decisions if additional zebra mussels are found, or in other water bodies when zebra mussels might be found or would be of concern. And the ones that I want to highlight were the development of a molecular probe for zebra mussels, looking at adult and larval growth and survival, in particular in relationship to calcium concentration as well as pH within a water body. We don't know, but of interest would be any potential impacts of increasing salt and other de-icing agents, as this is more widespread, and what potential impacts in synergy or combination with other exotic species would be. We had included a manuscript, Frischer et al., 2002, that hopefully you have access to. This was the development of a probe, a zebra mussel specific probe, in order to potentially facilitate the monitoring program and sensitivity of detection of zebra mussel veligers. This is just a figure from that paper, and we could develop a quagga mussel specific probe as well, because I realize that that's of major concern in a lot of areas. This has implications potentially for eDNA and use with the boat wash stations, because one limitation of the boat inspections to date is that they're not doing microscopy and looking in the water itself. They're looking for something that can be seen visually, such as the adult mussels, but we may be missing knowing when veligers are being brought in. So that we feel that this can be a very useful early detection tool in a wide variety of instances. As mentioned, looking at calcium as well as in different water bodies and the impact that that has, we wanted to experimentally carry out some bioassays and ascertain what survival percentages of adult zebra mussels were in Lake George versus in other water bodies. We selected the Hudson River and then made a control comparison in artificial water. Probably of most importance here or interest was that actually the zebra mussel survival, adult, let me say, in Lake George was 85%, which was a bit higher than in the Hudson River. However, if we look at the shell length, so the growth as well as the weight, clearly the zebra mussels in the Hudson River water were more robust. But the takeaway is that the adults can certainly survive and grow in Lake George water. We also wanted to look and see whether or not the veligers could survive within Lake George water, and this was a simple experiment where we placed veligers in glass slides for them to settle onto for 9 days in either Lake Champlain water or Lake George water, and take away the veligers did not survive in Lake George water. That is your typical Lake George water, not in a particular niche or area where calcium would be elevated. And finally, we did manipulations to see whether or not, if we mimicked both the pH and calcium concentrations in Lake Champlain water, would we see a similar response in terms of the survival of veligers. And so the blue is Lake Champlain, orange is Lake George to mimic it, and the gray is Lake George itself. And we see in Lake George water it is pH and calcium that's having a major impact on the death of the villagers. So to conclude, our lessons learned and recommendations. It's critical, as I'm sure you know, for there to be early detection as well as continued monitoring. Stakeholder involvement is also really essential. Having a response team and action plans are very important. Prevention and eradication, at least we have an example of a localized eradication as possible, but it certainly helps that in Lake George's instance, calcium concentrations were suboptimal. Bioassays are very useful, and in general, be prepared for surprises. If it's not zebra mussels, it could very well be the next invasive species. Marc and I need to thank, and this will be in the webinar, so you'll have it, but this would not have been possible with a tremendous amount of funding and efforts and collaborators, and we're grateful to all of them, as well as thank you for listening to our webinar today.
Thank you, Sandra and Marc. I think Marc had to leave us unexpectedly.
I'm still here. I got about 2 minutes left.
Okay, well, since you're still with us, maybe we'll take just that minute then. Although it looks like you've been answering the questions in the chat box, so I would just encourage folks to look at that chat box to see the questions that have been asked and answered, unless there's anything you want to add, Marc, on either of those questions.
No, Michael McCartney asked about whether the villagers, what might have happened with the villagers. Why didn't they survive? Did they just not metamorph, or did they actually die? We actually, in those experiments, were done in like aquarium tanks. We couldn't find the villagers anymore. It appears that they actually dissolved. So the calcium was so low that the villagers couldn't even maintain or couldn't make a shell, and they just disappeared.
Great. Well, thank you for that, and thank you both for presenting. Marc, I know you have to leave, but if Sandra, you can stay with us, we might have some more questions come in the interim, but I want to make sure we give our next two presenters enough time to get through their presentations. So I'm going to move on and introduce Dr. Brenda Lafrancois and Dr. Harvey Bootsma. Dr. Brenda Lafrancois is an aquatic ecologist for the National Park Service Midwest region and is stationed in Ashland, Wisconsin. Her work covers a broad range of aquatic issues, but frequently addresses effects of invasive species, particularly invasive mussels. She has worked on invasive mussel prevention, rapid response, monitoring, and research in systems ranging from large rivers and inland lakes to the Great Lakes. And since 2006, she has helped coordinate a collaborative research project addressing invasive mussel impacts at Sleeping Bear Dunes National Lakeshore in Lake Michigan. Dr. Harvey Bootsma is an associate professor at the University of Wisconsin-Milwaukee School of Freshwater Sciences. He conducts research primarily on large lakes, including the North American Great Lakes, lakes in South America, and the Great Lakes of East Africa. His work focuses on the response of aquatic ecosystems to external stressors, including invasive species, climate variability, and land use. Current research projects are addressing the effects of quagga mussels on nutrient dynamics and food web structure in Lake Michigan, and carbon dioxide dynamics in large temperate and tropical lakes. So Brenda and Harvey, you should be all set to get started.
Great, thanks for the introductions, and thanks so much to our fellow presenters, Sandra and Marc. That was a really interesting and encouraging presentation on the Lake George results, and I think everybody will find both some similarities and some differences in the case study that we'll present here today. So I want to start by introducing you a little bit to our study area and explaining how we came to be involved in mussel removal at all. The picture you're looking at here is from Sleeping Bear Dunes National Lakeshore, which is a unit of the National Park System situated in northern Lake Michigan in the state of Michigan. It's a really beautiful place full of big dunescapes and sparkling waters. These are the kinds of sweeping vistas and clean beaches that so many people come to the park each year to see. But in the last decade or so, there have been a lot of ecosystem changes occurring in Lake Michigan, and visitors have sometimes encountered these kinds of vistas on their beach walks. And this has happened as nuisance algal growth has really resurged in the lake and sometimes piles up on shorelines. Additionally, we've been seeing bird die-offs due to avian botulism outbreaks. These have become kind of an annual occurrence at the park, with hundreds or even thousands of dead water birds washing up on the lakeshore's beaches in some years. And in the cases of both nuisance algal growth and avian botulism outbreaks, the mussel invasion may be at the root of the problem. Zebra mussels were present in the park in the 1990s, but the big changes really happened after the quagga mussel invasion occurred, more in the 2000s. And by the mid to late 2000s, there's pretty much a blanket of quagga mussels over the whole lake bottom at Sleeping Bear Dunes, even at substantial depths. So in response to all of those issues, some time ago we initiated a bunch of intensive research on nearshore ecosystem changes. This is in collaboration with our partners, Harvey from the Milwaukee School of Freshwater Sciences and other partners. By 2010 or so, we'd begun to focus efforts on a particular site, and that's shown here is Good Harbor Reef. It's where the arrow is pointing to in this slide. And we picked that site for its proximity to a key marina, as well as its ecological and fisheries significance, and its representativeness of other local reef ecosystems. And so at that site, we set up some continuous monitoring devices, and we've also been exploring a variety of research themes at that site, including things related to nearshore food web structure and dynamics, changes in nutrient energy dynamics in the nearshore and offshore, and development of a nearshore ecosystem model. A larger scale, longer term objective is to develop a nearshore ecosystem model that can be linked into a whole lake model and help guide decisions related to phosphorus and fisheries management. In any case, over the course of this work, we've also conducted periodic bottom sampling at the Good Harbor site. And that bottom sampling consists of what you see in this slide, which is scraping mussels and related biota off of rocky surfaces. And what happened was that after a few years of sampling at that site, we began to notice that mussels were not recolonizing these scraped areas. In fact, there was so little recolonization that we actually had to move our monitoring station to a new site in order to have enough material to sample on an ongoing basis. And this observation that mussels did not recolonize the scraped sites was really the genesis of the invasive mussel removal experiment that we'll be talking about. So with that, I will turn it over to you, Harvey.
Okay. So as Brenda mentioned, we noticed that mussels weren't recolonizing these sites. Up until this point, a lot of our work had focused on what was causing the excessive growth of this nuisance algae on the lake bottom, which we believed was mussels were playing a role in. And we were looking at the effect of mussels on trophic dynamics and food web structure in Lake Michigan, and also looking into the botulism problem. But then in 2016, we thought, well, instead of just monitoring what's going on out there, let's do some experiments to gain some better insight into what the mussels are doing and how the system might respond if we didn't have mussels. So we decided to do this mussel removal experiment, and there were several questions that we wanted to address with this experiment. One was the question that Brenda already pointed out, was do mussels recolonize areas after they've been removed? We had evidence from our previous work that they weren't recolonizing scraped rocks, but we wanted to do that at a slightly larger scale. And then along with that, we wanted to see how the rest of the primarily benthic community also responded, because our hypothesis is that mussels are important recyclers of phosphorus, and that it was the recycled phosphorus from mussels that was driving the growth of this nuisance algae Cladophora. We were also interested in how the invertebrate community would respond, because that's an important food source for a lot of the nearshore fish community. and also interested in the microbial community. Brenda mentioned the problem with avian botulism in the Sleeping Bear Dunes area, and we have hypotheses that mussels and nuisance algae and round gobies are playing a role in the growth of the bacteria and the transfer of that toxin into the food web. And partly because of that, we are also interested in what role the mussels are playing in controlling the community composition with regard to fish. And this partly relates back to the botulism issue, because most of the birds that have been positively identified with botulism have had round gobies in their guts. So in 2016, we started removing mussels, and it turned out to be, I think, a bigger job than we thought it would be. We initially were naive and thought it would be kind of like going in and shoveling snow off the driveway, but it was more tedious than that. It's a very heterogeneous bottom. It's very lumpy. It's not a smooth bottom. And so to really effectively remove all the mussels, it took the divers quite a while, just going down with scrapers and scraping them off the rocks. So we did that throughout the summer of 2016, starting in late spring and going right to fall. And that was done both with the dive team here from UW-Milwaukee that went up a number of times, but we also had the help of quite a few volunteer citizen divers who were also going out there frequently and just doing as much scraping as they could. So here's just a couple bags. The previous talk mentioned that 21,000 or so mussels were removed from Lake George. We removed a few more than that from Lake Michigan. At the end of the summer, we had close to 40 square meters scraped, and we estimated that that was close to 1 million mussels that we had scraped off. And when they were scraped off, they were put in duffel bags like these and then transported away from the experimental site. So here's a site or a picture of the work in progress. So you can see these yellow floats. They demarcate the perimeter of the area that we've scraped. And this is partway through the summer. So you can kind of see a different color on the rocks that have been scraped. They don't have as much of the green algae on them as the rocks around them, and all the mussels have been removed from those rocks. And you can also see some of the monitoring equipment that we put up at this location. On the far left at the bottom, there's a YSI sonde to measure water quality. And then in the gray cylinder you see there, we had a time-lapse camera so that we could collect frequent images of the experimental area. The camera's set up to collect images every half hour, and then we put that all together later in a time-lapse video. And that gives us a good idea of how fast algae is growing on the site. It also allows us to do some monitoring, kind of semi-quantitative monitoring of fish, especially round gobies on the site. So as I mentioned, by the end of the summer, we had about 40 square meters cleared, around close to 1 million mussels. You saw in the previous picture the camera was on the edge of the cleared site, and here we're expanding that. So by the end of the summer, the camera was actually more like in the middle of the cleared area. And so that's what we did most of that summer, was just scraping. And then we started doing some monitoring towards the end of that summer as we got that whole area scraped. So I'll show you some graphs of the results, but here's just kind of a quick summary of some of the main observations that we made during this experiment. One thing we found was that in the spring and the early summer, the benthic algae are dominated by diatoms, and this was both on the scraped and the control areas. And this has been a little bit interesting because you saw in the previous image that rocks that we had scraped even several years ago, you could still see where the algae and the mussels had been scraped off of the rocks. And yet, when we started this experiment, we expected those rocks to look the same, and yet the following year in the spring, there was a lot of algae on the rocks that had had the mussels scraped off of them, but it was mostly diatoms. But as you progress through the summer, the diatoms give way to filamentous green algae, and then we start seeing less algae on the rocks that have had mussels scraped off of them compared to control rocks in the surrounding area that still have mussels on them. We also saw that the algae on the scraped rocks where there are no mussels had a lower phosphorus content, and that kind of supported our hypothesis that mussels are a provider of phosphorus to the algae and are helping to promote the growth of nuisance algae. And as I said, the scraped rocks, they remain predominantly diatoms, but the rocks with mussels on them, that algae gradually changed to filamentous greens dominated by Cladophora. We also did experiments looking at the excretion of phosphorus by mussels, and I'll show you one or two graphs that show how important the mussels can be as a phosphorus source. So here's some data from the first and second summer. So 2016 was when we actually scraped the mussels off. So the first data points there that you see in May, that was before we did the scraping. And the graph is showing biomass of algae on these rocks. So lots of algae there. And then the orange line there, the brownish orange line, is the algae on the rocks that we scraped the mussels off of. And so obviously in the first summer in 2016, that really dropped after we did the scraping because we removed the algae as we were scraping the mussels off. So the second year, 2017, that data is more meaningful. And there you can see, again, at the beginning of the summer, algae biomass was high both on the scraped rocks and on the control rocks. And then as we got into the summer, the algal biomass on the scraped rocks was lower than it was on the rocks with mussels. But there was still significant amounts of algae growing on the scraped rocks. But as I said, the composition was different. In the top slide on the right there, the top picture, you can see that's predominantly Cladophora that was growing on rocks with algae and mussels. Whereas the bottom image there, that's from the rocks with no mussels, and you can see that's predominantly diatoms. And I mentioned also the change in phosphorus content. Again, if we focus on the second year on 2017, you can see that the phosphorus content in the algae on the rocks with no mussels, that's the orange-brown symbols there, was quite a bit lower than the phosphorus content in the algae on the rocks with mussels. That dashed horizontal line there is the line that, based on previous assays, is considered the threshold for growth of Cladophora in the Great Lakes. So those mussels on the rocks without, or sorry, the algae on the rocks without mussels, they're really hard pressed as far as their phosphorus content grows. They have barely enough phosphorus to be able to grow. This is the result of some experiments measuring phosphorus flux from the benthos. And I guess first look at the far right column there. That's simply showing the excretion rate of mussels on an aerial basis. So that's micrograms of phosphorus per square meter per hour. So it shows you the excretion of dissolved phosphorus by mussels. And then if you look at the bar on the far left, that shows you chambers in which there was both algae and mussels. So the mussels are excreting at the same rate in those chambers, but the amount of phosphorus released into those chambers is lower because the algae is taking up a lot of the phosphorus that's being excreted by the mussels, and that's shown in that middle bar there. So what this tells us is that mussels are an important source of phosphorus, and that at least under these conditions, algae is taking up a lot of that phosphorus that is excreted by those mussels. This is just another slide showing some results from this year, from 2018. So we haven't plotted up all the data from this year, but we have some qualitative results here. Again, you can see in early summer, this is what the bottom looks like in early summer. So dominated by diatoms. You see all those white blobs on the rocks that look like cotton balls. Those are all filamentous diatoms that are growing on the rocks. And that was everywhere, both on the control site and on the rocks from which mussels had been removed. Here's some samples that we collected in our last trip just a few weeks ago to this area. So on the left you can see algae from the control area, that's with rocks with mussels on them. And on the right you see algae from the scraped area where there are no mussels. And there's a couple of differences you can see here. There's more biomass in the control sites. So the presence of mussels does seem to be promoting the growth of the algae. Also, you see the differences in color. That's partly due to the fact that algae on the scrape site are dominated more by diatoms, which tend to have a brownish yellow color, but it's also due to a difference in chlorophyll content of the algae. Here you can see a couple of photomicrographs here of Cladophora from the two different sites, and you can see that the figure on the left, there's a lot of chloroplasts in that filament, whereas the figure on the right, which is Cladophora from the scrape site, very few chloroplasts in that filament. So again, it looks like the algae that's growing on the rocks where the mussels have been removed is being stressed with regard to nutrient supply. And Brenda, I think you're going to take it over from here again.
Yes, that's right. Thanks, Harvey. I think that gives a good overview of the potential ecological outcomes of removal activities. And we wanted to leave everyone with some considerations and perspectives related to manual removal efforts, as well as some other potential applications where it might be effective. So, one and maybe the most obvious consideration relative to manual removal efforts is that it is fairly labor intensive. As Harvey noted, a lot of divers are needed. I think that was also noted in the Lake George presentation, although we note that divers are also likely needed for other types of removal methods. Because of that level of effort, the scale at which the treatment can be applied may be limited, and looking at the responses of certain kinds of biota like fish would likely require larger scale removal efforts. And one thing we wanted to note is that we've found the pre- and post-treatment monitoring to be really critical to understanding, what the outcome of the removal is from an ecological standpoint. And so we've made a lot of use of high-frequency monitoring with cameras and in-situ sensors to help get a handle on that response. From a long-term success type of perspective, a lot of the things we've noted here overlap with those in the Lake George presentation, but key to understand that success probably depends a bit on context. In the case of our work in Lake Michigan, we're finding that community composition matters because it appears that after mussel removal, round gobies likely provide a lot of biocontrol that helps ensure longer-term success. A related note is that depth and substrate matter because predation by those round gobies is likely higher at shallower substrates or shallower depths and on rockier substrates. And then, as was noted earlier, it matters whether or not there's a lot of propagule pressure. So the odds of mussel reestablishment are probably lower if there aren't a lot of new introductions coming from surrounding populations or in a riverine context from upstream populations or other sources. And then likewise, habitat concerns are worth considering because the odds of mussels reestablishing may be lower at sites that are kind of marginal habitat where there's lowish or medium calcium concentrations, marginal food quality, low temperatures, and so on. In other words, where their populations are not currently thriving. In terms of benefits and drawbacks of manual removal relative to other methods, one big benefit is that essentially you get a reset of the substrate if the mussels and shells are entirely removed from the site. And along with that comes the fact that you don't get any confounding issues associated with mussel decomposition. Another benefit is that it can be performed in an open water context without the use of barriers or any kind of complicated technology. It's got some simplicity to it, and along with that comes minimal permitting issues. Some of the limitations are, large labor input, small scale of applications, and the fact that it can be difficult to get access to the required dive expertise and boats. And so as Harvey mentioned earlier, one of the ways that we've tackled that is through a citizen dive program. And just to give people an idea of the mechanics of that, it essentially has involved developing an MOU That involves the Park Service, UW-Milwaukee, a local college, Northwestern Michigan College, and a local dive shop, Scuba North. And a lot of the dive coordination and support comes from that collaboration. And a lot of the financial support for the Citizen Dive program, which supports boat rentals and things like that, has come from Great Lakes Restoration Initiative and National Parks Conservation Association. And then we've also developed a close working relationship with a fairly local fisheries research station of the Michigan DNR, and they maintain a roster of trained scientific divers as well who have been able to contribute to the project. The application that we've talked about here is essentially, on a highly infested Great Lakes reef kind of context, but I thought it might be worth mentioning a couple other potential applications as the Park Service is involved in other kinds of manual removal efforts in the Great Lakes region. So one of these potential applications involves high-risk locations, docks and marinas, and at a couple of Lake Superior parks like Apostle Islands and Isle Royal, these types of situations are routinely monitored, and when we find zebra mussels present at those sites, they are manually removed. And so you can see these in this kind of context, we're talking about high risk but fairly low density, so we can just kind of pull them off as we see them. Another potential application is in cases where there are high value resources. So an example of that might be a shipwreck. At Apostle Islands, there's a steel-hulled shipwreck called the Savona that is lightly infested that we have been gradually removing mussels from. And another example might be a threatened or endangered resource like a native mussel resource. And for example, on the St. Croix River, zebra mussels have been experimentally removed from native mussels to help facilitate their recovery. And there's one other application here in terms of isolated populations. This is a similar situation to the one in Lake George, and a very recent one where an isolated population of zebra mussels was encountered at Isle Royal National Park in Lake Superior near a dock setting, but the population had been missed previously during surveys, and was a fairly dense but fairly localized grouping of zebra mussels. And over the last 2 weeks or so, about 2,300 mussels have been removed, and they are still involved in that effort. So these are just a few of the situations in which manual removal might be a viable option, and there are probably others within your management purview where these kinds of things could work. I guess I wanted to note finally that manual removal can also be paired with other types of control technologies, so it could be used following application of other treatment options to remove mussels and shell material from the site. And it can also be used as part of just follow-up monitoring and long-term maintenance and control following other kinds of control efforts. So that's all we have. I just want to acknowledge the many, many people who have contributed to the control effort at Sleeping Bear Dunes, which include our citizen divers and our research partners from Michigan DNR, University of Michigan, and Northwestern Michigan College, and most of all, the Great Lakes Restoration Initiative, which has made a lot of that work possible. Thanks.
Thanks, Brenda, and thanks, Harvey. Those are great presentations, and that concludes all of our presentations for today. I think we are running a little over time, but I haven't seen a great drop-off in webinar participants, and if you guys are willing to hang on for a few minutes, we can take some additional questions at this time. So the first question that we got for Brenda is the population at Isle Royal thought to be from natural reproduction or translocation from watercraft?
It is unknown at this point. It's because of its location, transportation via watercraft is suspected.
And also, Brenda, Harvey, and Sandra, if you guys have questions for each other, feel free to ask those as well. And webinar participants, as a reminder, you can type your questions into the chat box of the webinar window. Brenda, a follow-up question on the Isle Royal population. Is there more than one cohort there?
We're in the process of measuring them right now, but it kind of does look like there's one main cohort at least.
And another question probably for Brenda or maybe Harvey. What is the estimated size of Good Harbor Reef?
Oh.
I'd say it's roughly close to 10 square kilometers.
Probably a good guess.
Can I ask a question related to Lake George?
Yes, please.
I was wondering what the substrate is like around the lake, because we know especially zebra mussels are really limited by substrate. They need a hard substrate. So I was wondering if most of the nearshore is hard substrate or soft substrate, and if that's related all to the distribution of mussels that they're seeing around the lake.
So the substrate does change. Right now we also have Asian clams that have really taken a hold in the sandy areas. The zebra mussels that we were removing, a lot of people build their seawalls out of concrete, which is not great. And we have pilings for the docks. We do have, though, other areas that are rocky, or pebbles, and those have provided good substrate. We have a lot of lumber still left over from when there was harvesting of the forests, so that provides them with a good hard substrate as well. So it's kind of a mixed bag. But in the example of the area that Marc was talking about down in Lake George Village, part of the problem there was that a requirement in doing the construction of the boardwalk was to preserve the habitat for the fish. And so they put in a bunch of rocks back in that area for the fish, and those were really challenging to be able to get the zebra mussels off of. So it's a mixed bag.
I have a related question about substrate. I heard during your presentation, I think this was during Marc's portion, that in some cases soft substrate kind of made it complicated to look for the mussels where you were trying to look for them. And I wondered if you had found any in or on the soft substrate. And I raised this because we had an observation last year from Apostle Islands area in Lake Superior, and it was an area of very soft substrate, clays and silts, and we had confirmed zebra mussels living on that substrate, and that was the first time I had ever seen or heard of them doing that.
We haven't seen a lot of zebra mussels on soft substrate, although we have seen them attached to some of the submerged aquatic plants. Which is another potential mode for their transport, clearly, to other locations. We have some kind of sandy, gravelly type substrate where we would also find them attached, but to a silt or even a clay, we have not seen that.
Thanks.
And Brenda, I have another question, I think, for you. The question is on the St. Croix example about removing invasive mussels from native mussels, if we can provide a contact for that or if there are preliminary results from that work.
Yes. Yes, contact me directly and I can tell you more. I wish we had better results, but there were some complications with the experimental setup that year that made it hard to understand what the outcome of that work was. But yes, I can tell you more, and it is just a general matter, of course, in a lot of the mussel monitoring, native mussel monitoring activities on that river to remove invasive mussels in the process.
Okay, so Laureen is the person who asked that question. We will follow up with her and provide your contact information to her. And I believe we also have a couple of reports and papers related to this issue, manual removal of mussels from native mussels, that we'll be distributing with the webinar recording and made available on the Mussel Collaborative website as well. So those will also be available. And so, Brenda, I have another question for you. Have quagga mussels been confirmed from Apostle Islands? And if so, is it thought that they are reproducing?
Yes, and if Mike McCartney is still on, he might be able to help me with this one. In almost all the ones that we've confirmed so far have been confirmed zebra mussels, but there were, I think, 2 quagga mussel juveniles confirmed from a certain area in the park, and it's not clear if they were reproducing or not.
We don't see Mike in the participant list anymore, so he may have had to leave. All right, I am not seeing any more questions come in, and like I said, I think we are over time, so I am going to go ahead and wrap up for the webinar today. I want to thank all of our speakers and our participants. I also want to acknowledge the U.S. Geological Survey and the Great Lakes Restoration Initiative for providing funding support to the Invasive Mussel Collaborative. And as a final reminder, the webinar recording will be posted on the collaborative website in the next day or two. And if you have additional follow-up questions or comments, please feel free to contact us at the Great Lakes Commission. Thank you very much, everyone. Have a good day.
Thanks.
Thank you very much.